My name is Fatina Shada binti Anarhaq, metric number 17087-808. So in this video, I will explain to you more in detail about cancer drug resistance. So let's go. So drug resistance is a well-known phenomenon that occurs when a disease becomes no longer susceptible or become tolerant to its pharmaceutical treatments. This concept was firstly discovered when bacteria become resistant towards certain antibiotics. However, unfortunately, these similar mechanisms have been found in other diseases, including cancer. So, my question is, why do we have to study the mechanism of drug resistance? There are several needs for studying the drug-resistant mechanism of cancer. Firstly, to provide rational therapy. A more effective and suitable solution can be given to the patients in curing the disease. Next is the development of new drugs. This development of new drugs can overcome the drawbacks from the drug-resistant mechanism of cancer. And lastly, maximum chances of failure treatments can also be excluded. Therefore, mechanism of cancer drug resistance should be studied. So, these are the hallmarks of anti-cancer drug resistance. Firstly, drug inactivation. Next, alteration of drug targets. Drug efflux pump. DNA damage repair. Cell death inhibition, and lastly, cancer cell heterogeneity. Drug activation is in vivo process where the agent interact with different kind of proteins. The interaction leads to the modification, degradation, or even complex formation with protein or other different molecules, which eventually leads to the activation of the drugs. Anti-cancer agent may go metabolic activation, which resulting in acquire of clinical efficiency. However, cancer cells that produce resistance to this mechanism will lead to the decrease in the activation of the drugs. One of the examples of drug inhibition is acute myelogenous leukemia with cytarabine, RRC. This drug is activated after multiple phosphorylation, which results to RRC triphosphate. Downregulation or mutation leads to decrease in the activation of RRC in the pathway, which eventually leads to produce RRC resistance. Next is the alteration of drug target. The molecular target is most important factor for determining the efficiency of the drugs. The alteration of these targets by modification or mutation leads to the failure of the treatment by anti-cancer agents. In cancer, this type of alteration can be one of the causes of drug resistance. Third is efflux pump. Drug efflux pump is one of the most important and well-studied mechanism of drug resistance where the cancer cells do not let the drug to accumulate in the cell at desired concentration to its effect, which lead to the failure of the therapy. The ATP binding cassette protein transporter or ABC transporter is responsible for the efflux of the drugs and is well known studied regulated in human plasma membrane of healthy cells. So what is ABC transporters? ABC transporters are transmembrane protein which is present in healthy cells as well in large extent of cancer cells whose mechanism is to transport different type of substance to cross cellular regions. One of the example is estradiol. Estradiol is used in the treatment of breast cancer. It is the most effective drug but have been resisted due to the efflux pump mechanism of cancer cells. Next is DNA damage repair. The DNA damage repair mechanism has the vital role in anti-cancer resistance as this mechanism can produce drug resistance effect on chemotherapy. the damaged DNA repair mechanism of the cells can actually reverse the damage produced by the drug to the cancer cells in chemotherapy and lead to produce its resistance. For example, platinum-containing chemotherapeutic agents like cisplatin, which cause harmful DNA cross-link and eventually leads to apoptosis. The resistance of these drugs can arise due to nucleotide homologous recombination and leads to the repair of the primary damage of DNA and also the failure of the treatment of the cancer drugs. Therefore, drug resistance will produce and the response of the anti-cancer agents will also inhibit. There are two main mechanisms that were involved in cell death inhibition, which are apoptosis and autophagy regulatory events. Both of them are acting antagonistic to each other. Apoptosis consists of two distinct pathways, which are intrinsic pathway and also extrinsic pathway. In the intrinsic pathway, which is associated with the mitochondrial beta cell lymphoma 2, or well known as BCL2, cascade 9, and also protein kinase B, AKT. While for the extrinsic pathway, where the death receptors are involved on the surface of the cells. Both the intrinsic and extrinsic pathways get activated when there is the increase in the level of the cascade 3 and also AKT, which ultimately lead to the apoptosis of cell. For instance, fluorouracil in combination with chloruracil had greater effect for cell death mechanism, which has been inhibited by the cell death inhibition mechanism. And lastly, cancer cell heterogeneity. In this mechanism, the cancer cells do increase in growth, but already have the resistance toward the anti-cancer agents. Recent studies showed that heterogeneous cancer cells has the stem cell properties, which enables it to produce cells with anti-cancer resistance property. So that concludes all 6 drug-resistant mechanisms of cancer. And that's all from me. Thank you for listening.